Hello everybody again. Now we are going to continue to our lecture about the eye histology from the vascular layer. As we talked about that, vascular layer composed of the choroid in the outer part, in the posterior part, ciliary process, and the iris. Choroid in itself have a posterior area known as the choroidocapillary lamina. And this choroidocapillary lamina, we have high amount of blood vessels, which have a role in nourishment of the distal parts of retina and also retina pigment epithelium. In the inner side, we have a layer neighbor to the retina pigment epithelium. This layer known as the broad membrane. The broad membrane composed of the basement membrane of retina pigment epithelium and also the basement membrane of the blood vessels which present in the choroidocapillary lamina. Broad membrane actually have a role in blood retina barrier or blood retina pigment epithelium barrier, and blood in the inner side of the capillary of choroidocapillary plexus connected with the broad membrane and transfer some materials to the retina pigment epithelium and also give waste structures from that. The ciliary body present between the iris and choroid. The ciliary body have the ciliary muscles which have a role in accommodation, and also we have the ciliary process. The ciliary process covered with two layers of epithelial cells. These two layers of epithelial cells have a role in production of ciliary flow. Okay, look at here. This is our ciliary process. This part is our posterior chamber area. And this area is of the connective tissue area. Here you can see two layers of epithelial cells. These two layers of epithelial cells have two different structures. This layer, which look at the posterior cavity, known as the non-pigmented epithelial layer or inner non-pigmented epithelial layer. Why I call inner? Because present in the inner side of the eye and near to the posterior chamber. And in outer side of this epithelium, non-pigmented epithelium, we have another epithelial layer. This layer is pigmented epithelial layer or after pigmented epithelial layer. And here we have connective tissue filled with blood vessels. Between the blood vessels and epithelial layer, we have another barrier, blood ciliary body or blood, I forget the other name of this barrier, the barrier between the blood and these cells, the pigmented epithelial cells, and this process have a role in production of fluid and also have a role in production of zonular fibers which connected with lens. These fibers connected with this non-pigmented epithelium from one side and from other side connected with lens equatorial region. The epithelial cells or non-pigmented epithelial cells in the inner side of the ciliary epithelium produce the aquasomor. This aquasomor released to this part and then goes from the pupilla to the anterior side, and after that continue with scleral venous sinuses or Schlemm channel. We talked about aquasomor. Ciliary zones. Here you can see the ciliary zonulas connected with the lens and ciliary bodies. The other structure in the end of the vascular layer of eye, known as the iris. The iris have two areas, the anterior and posterior side. In anterior side of iris, we have structure like connective tissue without epithelium. This side of iris. This side of iris. And we have posterior side of iris covered with epithelial cells like ciliary process epithelium. But here we have a differences between the ciliary process epithelium and iris epithelium. In iris, again, we have two layers of epithelial cells. But these two layers of epithelial cells, or both of them, is pigmented epithelial cells. You can see non-pigmented epithelium here. Non-pigmented epithelium appear in the inner epithelial part of the ciliary process. In iris, we have two layers of pigmented epithelial cells. Okay, this is our iris section. This part is anterior chamber, this part is posterior chamber, and this is our lens. This part is anterior side of the iris, and here is posterior side of iris. In anterior side of iris, we have connective tissue filled with melanocytes. The number of melanocytes and melanin pigment in this connective tissue related with the color of eye. If the amount of these cells high in the iris, we have darker eye color, and in persons with light eye color, we have low amount of pigmented cells in the connective tissue. Okay, the amount of cells in connective tissue related with the eye color. Can you understand? In iris, also we have two distinct layers of smooth muscle cells. The sphincter pupilla muscle and dilator pupilla muscle. This is our sphincter pupilla muscle and this is our dilator pupilla muscle. And these muscle layers have a role in increase and decrease the diameter of pupilla. Okay? With contraction of sphincter pupilla muscle, the diameter of pupilla is decreased, and with contraction of dilator pupilla muscles, the diameter of pupilla is increased. And in the posterior side of iris, we have this pigmented area or pigmented epithelium. This pigmented epithelium composed of two layers of the pigmented epithelial cells. Guys, the pigmented epithelial cells in iris, in ciliary body, and retina pigment epithelium continue with each other. Actually, these cells continue with retina pigment epithelium. That cells goes to the tip of the iris and then turn in this part and again continue into the inner side. With this turning and continuing, the pigment epithelial cells again present in this part, but this pigment and pigment granules goes to disappear in the ciliary process inner epithelium. Ciliary process inner epithelium continue with the retina pigment epithelium. We don't have embryology lectures for dentistry students. In embryology lectures, we teach this part and the formation of these layers, but for you, we don't teach this part. And if you want, you can read these parts for yourself and understand the formation of these layers. Do you have questions related with iris? Here you can see high magnification of iris, and this is our stroma. And here you can see sphincter pupilla muscles and dilator pupilla muscles near to the pigment epithelium. Okay. The other topic is lens. What is the structure of lens? Lens totally covered with a capsule from outer side. And under the capsule, we have the lens epithelium, simple cuboidal epithelial cells present under the lens. But the lens epithelium or subcapsular epithelium present only in the anterior surface of lens, and in posterior surface of lens, we don't have this epithelial layer, and lens directly connected with the capsule. These cells goes to the equatorial ridge of lens and then start to mitotic division and transform to long cells. Here you can see these long cells. And after complete the differentiation, the unmature cells or the cells in differentiating stage lose their nuclei and transform into the lens fibers or mature lens fibers. And the nuclear region of cells appear like these empty embryos, or in some references they call ghost cells to the cells of the mature lens fiber embryos. Now let me talk about the accommodation, how the diameter of lens increase and decrease. When we're going to look at the objects in distance, the lens need to flatten. For this function, the ciliary muscles relax. With this relaxation, the zonulary fibers distended, and the lens which connected with the zonular fibers goes to the equatorial area and lens goes to flat. When we going to look at the objects in closed or enclosed area, the ciliary muscles contract. With this contraction, the zonular fibers relax. And with this relaxation, lens again goes to rounded and increase the thicknesses of lens. Can you understand the physiology of accommodation? Why the relaxation of ciliary muscle lead to decrease the thicknesses of lens? Why? And why the contraction of ciliary muscle increase the thicknesses of lens? This is our ciliary process. And here we have the ciliary muscles. And this ciliary process connected with lens. Okay? When these ciliary muscles contract, the structure of ciliary process goes to close into each other. Like this. And with this closing in the structure of ciliary muscles and ciliary process, our ciliary process give a form like this. And the length of ciliary processes increase with contraction. When you contract the ciliary body from this part, the length of these processes increase. Because of this, the zonulary fibers relax and the lens goes to increase the thickness. But if these muscles goes to relaxation, the tonus of the muscles decrease, and with this decretion, the length of ciliary process decrease. For example, we have a ciliary process like this. Because of the relaxation of muscles. With this relaxation, the length of ciliary process decrease, and this decrease lead to tension of the zonulary fibers. With this tension, the thicknesses of lens is decreased. Can you understand? Okay. We talked about vitreous body and hyalocytes. And now we are going to talk about the different layers of retina. It's a very important layer. Okay. In retina from outer to inner side, in outermost side of retina, here we have the pigmented epithelium. And this cells is our retina pigment epithelium, which continue with the ciliary process epithelium and iris epithelium. And here is our choroid plexus, and here you can see a large capillary which connected with the retina pigment epithelium. These pigment epithelial cells have a role in nutrition of the rest part of the retina. In retina, we have different cells, and these cells present in different segments of photoreceptor retina. Look at here. This yellowish area is our retina. And here is our choroid. When we take a space and look at in the higher magnification, you can see choroid in this part and retina pigment epithelium here. The rest part of retina, this part, is our photoreceptor retina. In photoreceptive retina, we have the rod and cone cells or photoreceptor cells, bipolar cells, horizontal cells, amacrine cells, ganglions, and Muller cells. The location of these cells, interaction of these cells with each other is very important. This part is our end piece of the retina, or innermost layer of retina. And this part is our outermost layer. We have a cell with the name of Muller cell, a supportive cell, present in all of these layers. The basement part present here, and apical part present in this. A long cell, and this long cell have a role in supporting of the different cells of retina. Okay? The rod and cone cells, our photoreceptor cells, present in this layer, near to the retina pigment epithelium. They have the after segment, neighbor to the retina pigment epithelium, the nuclei side and cytoplasmic part, and the inner segment. From after segment, these cells have a connection with the retina pigment epithelial cells. In the inner side, the axons of these cells connected with bipolar cells. Bipolar cells, dendrites, connected with the axons of the rod and cone cells. And these bipolar cells transfer the photogenic materials, or transfer the information related with light from rod and cone cells to the innermost ganglion cells. Ganglion cells present here. The axons of bipolar cells connected with the dendrite of ganglion cells, and this connection lead to transfer of information related with light from bipolar cells to ganglion cells. Then the light information transfer from ganglion cells via the long axons to the central nervous system, with optic nerve in this side. This optic nerve composed of axons of these ganglion cells. Okay? Can you understand the relation between the photoreceptor cells, or rod and cone cells, bipolar cells, and ganglion cells? Okay? These three different types of cells have a role in conducting of light information. Also, we have two other cells, the horizontal cells and amacrine cells. These cells is supportive cells. One of them present in a little outer side of the retina layer, neighbor to the area of the connection with rod and cone cells and bipolar cells. The other one present a little in the inner side and have a role in the support and control of the connection between the bipolar cells and ganglion cells. Can you understand these relations? This is the simplest relation, but we have different types of bipolar cells, we have different synapses in this part, but I don't want you to know this extra information because you're not a specialist related with eye. You're a dentist, okay? The segment of these different cells and the axons and dendrite of these cells and the stage of these cells lead to formation of different layers in the retina. Now, we're going to talk about that layers. Look at here. This is our retina different epitheliums. Here is our choroid part, and this is our sclera. And here you can see our optic disc. In optic disc area, we don't have photoreceptor retina. Okay. Look at here. This is very important for your exam. Maybe I will ask the name of these layers or the structures or cells present in these different layers. This part is our colloid layer. And from here to here is our retina area. This part is our vitreous body or inner side of the eye, and this part is our outer side of the eye. In outer side of retina, we have retina pigment epithelium. After that, look at here. Okay, from here, this side. This side is the layer of rod and cones layer. In this side, we have only the outer segment of rod and cone cells. Can you see this line? Only this line here. This line is the outer limiting membrane. Outer limiting membrane. Here you can see. This layer formed with junction between the apical portion of Muller cells and the rod and cone cells. Okay, outer limiting membrane. In the inner side of outer limiting membrane, we have this area, after nucleus layer. After nucleus or nuclei layer. Here you can see. In the outer nuclear layer, we have the nucleus of rod and cone cells. Only the nucleus of rod and cone cells. Yes, also here we have the cytoplasma of rod and cone cells. Okay? In this layer, we have one other cell, cytoplasma. Which cell? Remember that lung cell? Muller cell. Muller cell cytoplasma present in all layers. All layers. So, here we have the nucleus of rod and cones and cytoplasma of rod and cones and Muller cells. Okay? Don't forget. In a little inner side, here, we have outer plexiform layer, this layer. In outer plexiform layer, we have the internal segments of rod and cones, we have the the dendrites of bipolar cells. Look at here, bipolar cells, dendrites. And the dendrites of horizontal cells present in this layer. And also the cytoplasma of Muller cells. In a little inner side, we have inner nucleolar layer. In inner nucleolar layer, we have the nucleus of which cells. What do you think about the nucleus of cells in this part? Which cells nucleus you think can be present in this side? Bipolar cells, horizontal cells, amacrine cells, and the Muller cells. 4 cells. Nucleus presented in the inner nucleolar layer. A little bit deeper side, we have inner plexiform layer. In inner plexiform layer, we have the axons of bipolar cells, dendrite of ganglion cells, and dendrite of amacrine cells. Okay? Amacrine cells dendrite present here. Horizontal cells dendrite present here. Don't confuse this. In a little inner side, we have ganglion layer, another nucleolar layer. In ganglion layer, we have the ganglion cell bodies or pericardiums. The other layer in inner side is nerve fiber layer. In nerve fiber layer, we have axons of these ganglion cells. Axons of ganglion cells present here, and in the innermost side, we have inner limiting membrane. Inner limiting membrane. Remember, we have an outer limiting membrane here, and here we have inner limiting membrane. What is this membrane? Remember that lung cells again. Muller cells. Muller cells basement compartment compose this inner limiting membrane. Apical compartment have a role in formation of the outer limiting membrane, and basement compartment have a role in formation of the inner limiting membrane. Okay? Can you understand? It's very hard. To understand this part, you should memorize this picture, the location of these cells and the connections. If you memorize this part, you can understand the histology sections. For example, here, this part is our rod and cone cell layer. This part is our outer limiting membrane. This nuclear part is our outer nuclear layer. Here is our outer plexiform layer. This part is our inner nuclear layer. This part is outer inner plexiform layer, and here is ganglion layer, and this part is nerve fiber layer. Here you can see the outer limiting membrane. It's very simple if you can memorize and understand the location of the cells, relation of the cells, and the function of these cells. Okay, we talked about these parts. You can read this part for yourself, and here you can see the schema related with the rod and cone cells. Okay, thank you to your attention. Our lecture related with eye ended in this part, and we are going to continue with the histology of